Hi there, welcome to ShineTech's YouTube channel. In this video, I'm going to talk about apoptosis, necroptosis, and pyroptosis. So let's start by looking at apoptosis. So apoptosis is simply a pathway of cell death in which cells activate enzymes that degrade the cell's own nuclear DNA and cytoplasmic protein. So in this type of cell death, enzymes are activated which will degrade the nuclear DNA and cytoplasmic proteins. So the organelles, the protein organelles inside the cell are going to be degraded together with the nuclear DNA by the activated enzymes. Then that type of cell death is what we call apoptosis. So under apoptosis, the plasma membrane of the apoptotic cell, the plasma membrane of the apoptotic cell will remain intact. It won't be damaged. And then the apoptotic cell does not elicit an inflammatory reaction. So inflammation is not observed during apoptosis. And then apoptosis involves caspases. So caspases are involved under apoptosis. During apoptosis, caspases are involved. Then from there, apoptosis is also called programmed cell death. Okay, so is a programmed cell death. So there are two mechanisms during in which apoptosis takes place. Okay, so we have the first mechanism or the pathway, which is called extrinsic pathway, and then we also have intrinsic pathway. Okay, so let's look at extrinsic pathway. So extrinsic pathway of apoptosis involves a ligand which is coming from the outside the cell, and that ligand binds to a receptor, and that receptor will initiate a cascade of reactions which will lead to apoptosis. So, but before we look at that, let's first understand one thing here. We have what we call stress sensors, and we have what we call pro-apoptotic factors and anti-apoptotic factors. So stress sensors, these are sensors which are going to sense stress, and then after they sense the stress, they are going to activate pro-apoptotic factors. And then pro-apoptotic factors, these are the factors which are now responsible to initiate apoptosis. Okay, so in the normal condition, we have anti-apoptotic factors which inhibit, okay, which are in the same, which balance up with the pro-apoptotic factors, preventing the cell from undergoing apoptosis, okay, preventing the cell from dying. Okay, so the stressor, the stress sensors, we have the beam, bead, Noxa, and Puma. So those are the stress sensors which are going to sense stress. Okay, and then the pro-apoptotic factors, we have the BAX and P53. Then anti-apoptotic factors, we have the BCLXL, and we also have the BCL2, and we have MCL1. So these are the anti-apoptotic factors which are going to balance up with the pro-apoptotic factors, preventing apoptosis. Okay, now that we understand what stress sensors, pro-apoptotic factors, and anti-apoptotic factors are, let's look at now an extrinsic pathway of apoptosis. So during the extrinsic pathway, we have a ligand which is called Faz ligand from the extracellular, from outside the cell, which will bind. Okay, it's going to bind to this receptor here outside the cell on the cell membrane, which is called the CP95. So CP95 is going to be activated. So once CP95, which is called the death receptor, is activated, we are going to have what we call FAD. So FAD is going to be activated. So FAD is simply phase associated death domain. So it is phase associated death domain which will activate caspases 8 and 10. So caspases 8 and 10 are going to be activated. So we have caspases 8 and 10, which are activated by the FAD, to active caspase 8 and 10. Okay, so once they are active, caspase 8 and 10 is going to activate caspase 3, 6, and 7. Okay, so caspase 3, 6, and 7, once they are active, they are going to lead to activation of these enzymes, which are here. So these enzymes are phospholipases, proteases, and we also have endonucleases. So these enzymes are going to degrade the cell. They're going to they're going to kill the cell. For example, phospholipases are going to degrade the membranes of the organelles inside the cell. We have the proteases. We also have the endonucleases, which is going to degrade the DNA. Okay, it's going to degrade the DNA. It's going to cleave the DNA into each 200 base pairs. So for every 200 base pairs, these endonucleases are going to cut or the DNA. Eventually the cell is going to die. So that's how this process takes place under extrinsic pathway. Okay, so we also have what we call intrinsic pathway, where now those sensors and the pro-apoptotic factors now are involved here. So during intrinsic pathway, what we are going to see is that the initiator is not coming from outside. The activator of this cell death is inside. That's why it is called intrinsic inside the cell. Okay, so what will happen here is that we are going to have we have this BCL2, which is an anti-apoptotic factor. So this anti-apoptotic factor BCL2 is on the membrane of the mitochondria. So it prevents the dangerous chemical, which is called cytochrome C. The cytochrome C is found inside the matrix of the mitochondria. It prevents it from leaking out into the cytosol. Why? Because this cytochrome C will initiate apoptosis by binding to APAF1. Okay, so what will happen is that if the stress sensors, okay, have activated the apoptotic factors, what will happen is that we are going to have leakage of this cytochrome C into the cytosol. Okay, the apoptotic factors are going to form pores. In which cytochrome C is going to leak out. Now, once the cytochrome C is inside the cytosol, what will happen is that it is going to bind to APAF1. So APAF1 is simply the apoptosis activating factor 1. So it is going to bind apoptosis activating factor 1, so which is APAF1. And once these two binds, they are going to form what we call an apoptosome. So an apoptosome is going to activate procaspase 9 to active caspase 9. Now this caspase 9 is going to do the same thing as the caspase 8 and 10 did in the extrinsic pathway, where we saw that the lipases, proteases, and endonucleases were activated. So this caspase number 9, which is activated during intrinsic pathway, is also going to activate these enzymes, which will lead to degradation of the organelles inside the cell, including the nucleus itself. So that's what happens during intrinsic pathway of apoptosis. Okay, so this is just the same thing. So once the cell has now died, what will happen is that all the organelles inside there have been degraded. The cell is going to form what we call apoptotic bodies. So the whole cell is going to form small body structures which are called apoptotic bodies. So those apoptotic bodies, they are going to be engulfed by what we call macrophages. Macrophages are going to engulf these apoptotic bodies. So these are simply small bodies from a apoptotic cell. So this process takes place when we have what we call phosphodieselling. Phosphodieselling is simply these structures on the apoptotic bodies. So those structures there are what we call phosphodieselling. Phosphodieselling is going to bind. It is always found inside the cell, but once the cell has died, it is going to find on the membrane, found on the membranes of the apoptotic bodies here. So on the cell membranes of the apoptotic bodies, that's where it is going to be found. So these acts as what we call eat me signals. So these acts as the eat me signals. They are the ones which signals macrophages to come and engulf these bodies. So macrophages are going to recognize these eat me signals, and then they are going to engulf. They are going to phagocytose these small apoptotic bodies, and finally they are going to be eliminated from the tissue. Okay, so that's what happens under intrinsic pathway of apoptosis. So the same applies to this diagram which is here. We have seen this cell is undergoing apoptosis. Okay, and apoptosis is taking place, forming what we call apoptotic bodies. Now apoptotic bodies have what we call eat me signals. Then those eat me signals are going to signal phagocytes to engulf those small apoptotic bodies, and finally the cell will be cleared. Okay, so apoptosis again can be physiological and pathological. Okay, can be physiological and pathological. In necrosis, mostly it is always pathological. So under physiological situation of apoptosis, what happens is that under physiological situations, so we're talking about physiological situations. So physiological situations of apoptosis is seen when during what we call embryogenesis, okay, and development processes. Okay, so where removal of excess cells takes place. So we have removal of excess cells during embryogenesis and development processes. Okay, so for example, we have disappearance of web tissue between fingers and toes. Okay, so during embryogenesis, in the first place, the limb will appear like a pad like this, and then the cells which are in between when forming the fingers like this. So the cells which are in between here are going to undergo what we call apoptosis, so that the fingers are formed, toes and fingers are formed. So that process is a physiological situation of apoptosis. Then apart from that, we have elimination of cells after withdrawal of hormonal stimuli. That is seen under endometrial. Cells will break down during menstrual cycle. So during menstruation in women, the endometrial cells break down. Okay, they'll break down and are released during the cycle. So that is also an example of physiological situation. So it's a normal thing. Now let's look at pathological situations of apoptosis. So pathological situations is seen when during elimination of cells with damaged DNA. For example, tumor suppressor gene P53 triggers apoptosis in cells with damaged DNA. So if you have cells which have damaged DNA, we have this tumor suppressor gene which triggers apoptosis. So that kind of apoptosis is pathological. And then we also have killing of viral infected cells. So if the cell is observed, or it is a cell has a virus inside, so there will be a signal which will initiate apoptosis so that that cell dies. Then from there, we also have cell death induced by anti-cancer drugs. So anti-cancer drugs also induce apoptosis. So that is a pathological situation. So that's all about apoptosis. Now let's look at the second part, which is necroptosis. Necroptosis. So necroptosis, we have two terms here. We have necro, we're coming from necrosis, and ptosis, which is coming from apoptosis. So this there are features of both necrosis and apoptosis in this type of cell death. Okay, and it is programmed cell death. That's why it is called necroapoptosis. It involves necrosis and apoptosis. It is a programmed cell death. So the way it happens, we have TNF, okay, tumor necrosis factor, which is going to bind to a receptor on the cell. Okay, so this is the mechanism of necroptosis. In the first place, we are going to have TNF, which is tumor necrosis factor, which is going to bind to TNF receptor. Okay, tumor necrosis factor receptor here. So that receptor on the cell. Now, once the TNF binds to TNFR, what will happen is that RIP kinase is going to be activated. Okay, so we're going to see RIP kinase is going to be activated inside the cell. So RIP kinase simply means receptor interacting protein kinase. Receptor interacting protein kinase is going to be activated once TNF receptor binds what we call TNF. Okay, and then from there, the RIP kinase is going to phosphorylate MLKL. There will be phosphorylation of MLKL, which now this complex is going to lead to plasma membrane damage and inflammation. So we are going to have a plasma membrane is going to damage. This plasma membrane here is going to damage. So once the plasma membrane damage, we are going to have accumulation of water ions inside the cell. And then some are going to leak out of the cell, and these which leaks out of the cell are going to cause inflammation, and then finally this cell is going to die. Okay, so in the first place, this was programmed that we had TNF, which had bind to TNFR. That's why it is called necroptosis. Why? Because it is programmed, and the mechanism I've explained it here. Okay, so that's what takes place under what we call necroptosis. Then we have also what we call pyroptosis. Okay, so pyroptosis. So pyro simply means fire. So meaning this kind of apoptosis or this kind of cell death is going to cause fever. So it is going to cause fever. So it is associated with activation of a cytosolic danger sensing protein complex, which is called inflammasome. So an inflammasome is going to be formed here. Now, mechanism, how does this pyroptosis takes place? So in the first place, we have an antigen from a bacteria or a microbe, okay, or a foreign body. Now, this antigen is going to bind to a receptor, which is called toll-like receptor on the cell membrane there. So once that antigen binds, the TLR is going to be activated, and it is going to activate caspases 11, 4, and 5. So these activated caspases are going to activate what we call gasdermin D. Gasdermin D, and this gasdermin D is going to make pores on the cell membrane of the cell. It is going to form pores on the plasma membrane. So those pores, once have been formed, we are going to observe cell swelling. The cell is going to swell. Why? Because we are going to have accumulation of fluids from the extracellular fluid into the cell cytoplasm. And once that happens, we are going to damage the cell membrane. And once the cell membrane has damaged, what will happen? Some fluids inside the cell is going to leak out. When they leak out, they are going to cause an inflammatory reaction. So inflammation is observed here, and also in the necroptosis also is also observed, as we have already explained that. So this is the one of the pathways in which pyroptosis takes place. So now let's see another pathway. So we have another pathway. Here we had an antigen binding to a receptor. Now in this second pathway here, what happens is that the antigen will penetrate the cell membrane, and then from there it is going to bind to NLR. So this is simply node-like receptor. Node-like receptor will bind to the antigen, and then from there it is going to form what we call an inflammasome. So this inflammasome, it is will now activate or initiate activities or cascades of activities which will lead to apoptosis, which will lead to cell death. Okay, so the inflammasome is going to activate procaspase number 1. Okay, so we have procaspase 1, which is activated here. So we are going to have activated procaspase 1. Then this activated procaspase 1 is going to activate gasdermin. So gasdermin is now going to do the same activities as I've already explained in the first pathway. And it's also going to activate another thing here, which is called interleukin 1. So the active caspase 1 is going to activate interleukin 1 to active interleukin 1. So this active interleukin 1 causes fever. So it causes fever. So that's how that's where the name comes from, pyroptosis. Why? Because it causes fever. We have activation of interleukin 1, which now causes fever. And here also we have activation of gasdermin, which causes what? Which is going to cause swelling inside the cell membrane damage, and finally inflammation. So thank you very much for watching. Don't forget to like, subscribe, and leave your comment in the comment section below.